Assalamu alaikum. Good morning, everybody. Today we'll cover the last lecture of the respiratory system, and the title of today's lecture is Regulation of Respiration. The objectives of the current lecture are to identify the regions in the central nervous system that play important roles in the generation and control of basic rhythm of respiration. This is at rest, as well as forceful breathing during physical activity. Describe how the pneumotaxic and apneustic areas are related to respiration. List the factors that influence respiration. List the anatomical locations of chemoreceptors that are sensitive to changes in arterial partial pressure of oxygen, arterial partial pressure of carbon dioxide, and hydrogen that participate in the control of ventilation, and identify the relative importance of each in sensing alterations in blood gases, and also to describe how changes in arterial partial pressure of oxygen and partial pressure of carbon dioxide alter alveolar ventilation. And also at the end we have to define hypoxia and classify it in its four types. Let's start with the respiratory center. The rhythmic pattern of breathing is established by the cyclical neural activity from the respiratory center in the brainstem to the respiratory muscles that we studied in the first lecture of the respiratory system. The respiratory center is composed of several groups of neurons located bilaterally in the medulla oblongata and pons of the brainstem, and it is divided into three major collections of neurons. The first collection of neurons is the medullary rhythmicity area, this one. So it is located here in the medulla oblongata, located in the medulla and composed of upper or dorsal inspiratory area and ventral expiratory area. And then we have another area called the pneumotaxic area. It is located in the upper pons. This is the pons, so the pneumotaxic area is present in the upper part of the pons. The third area is the apneustic area that is located in the lower part of the pons. So we have three major collections: the medullary rhythmicity area with its inspiratory and expiratory areas, and we have the other two areas located in the pons, the pneumotaxic area and the apneustic area. Regarding the medullary rhythmicity area, the neurons responsible for inspiration, or we call them the inspiratory area, are in the dorsal medulla, while those responsible for expiration are in the ventral medulla. So here we have the inspiratory area and the expiratory area, and this is the ventral side in this figure, and here we have the dorsal side towards the cerebellum. The neurons from the inspiratory area terminate on the motor neurons that supply the inspiratory muscles, and we know that the main inspiratory muscles are the diaphragm and the external intercostal muscles. The basic rhythm of respiration is generated mainly in the dorsal respiratory group of neurons. So it is mainly generated in the inspiratory area. The dorsal inspiratory neurons, when they fire, they will cause inspiration. So when dorsal inspiratory neurons fire, inspiration, which is an active process, takes place. And when they stop firing, expiration, which is a passive process, will take place. And this is the pattern of breathing at rest. We said that inhalation is caused by contraction of the inspiratory muscles, while exhalation at rest is a passive process due to the relaxation of the inspiratory muscles, and the two main inspiratory muscles are the diaphragm and the internal intercostal muscle, the external intercostal. The roles of the medullary rhythmicity area in controlling basic rhythm of respiration during normal quiet breathing. This is the inspiratory area. It is active for 2 seconds, and this activity will be sent in the form of action potential through motor neurons to the diaphragm and external intercostal muscles, and this will lead to normal quiet inhalation. Again, we are talking about normal quiet breathing, which is the breathing at rest. Then the following 3 seconds, the dorsal inspiratory area becomes inactive. So there are no more action potentials delivered to the inspiratory muscles. Therefore, the diaphragm and external intercostals will relax, followed by the elastic recoil of the lungs, and this will lead to the passive process of exhalation, which is the normal quiet exhalation. Therefore, inspiration for 2 seconds is caused by repetitive inspiratory signals from the dorsal respiratory group, which is the inspiratory area, transmitted mainly to the diaphragm and the external intercostal muscles. The following 3 seconds, which is expiration, results from the elastic recoil of the lungs and the thoracic cage. The neurons of the ventral group or the expiratory area remains inactive. So it is almost totally inactive during normal quiet respiration, which is the respiration at rest. So no expiratory area involvement in the normal quiet breathing. The area is silent during normal quiet breathing. During forceful breathing, what will happen is that the inspiratory area will fire at higher frequency. So in addition to the diaphragm and the external intercostals, the accessory inspiratory muscles are recruited to increase the tidal volume and also to increase the respiratory rate, and this will lead to forceful inhalation. The overactive inspiratory area will also activate the nearby ventral expiratory area. This will lead to the activation of the expiratory muscles, which are the internal intercostal muscles and the abdominal muscles. And this will lead to forceful exhalation. So forceful inspiration is caused by repetitive inspiratory signals from the dorsal respiratory group, which is the inspiratory area, transmitted to the diaphragm, external intercostal muscles, the sternocleidomastoids, and the scalene muscles. Forceful expiration results from the contraction of the internal, intercostal, and abdominal muscles, because impulses from the inspiratory area will activate the expiratory area as shown in this figure. Now, let's know what are the functions of the other 2 areas that lie in the pons. First, let's talk about the pneumotaxic center. It is located in the upper pons, as seen here in this figure. It transmits inhibitory signals to the inspiratory area. So when it is active, it will inhibit the inspiratory area. It helps to turn off the inspiratory area, and therefore it controls the rate and depth of breathing. For example, when the pneumotaxic signal is strong, inspiration may last for as little as half a second. During normal quiet breathing, we said inspiration will last for 2 seconds. But if the pneumotaxic center that lies in the upper pons is active, it will inhibit the inspiratory area, so the firing duration of the inspiratory area will be only half a second, thus filling the lungs only slightly. So if we have very short duration of inspiratory area activity, this will cause only contraction of the inspiratory muscles for that duration, and this will lead to the low or slight filling of the lungs with air. When the pneumotaxic signal is weak, on the other hand, inspiration may continue for 5 or more seconds, because now the inspiratory area is not under the inhibition of the pneumotaxic center. Therefore, the lungs will fill with a great amount of air and the tidal volume will be high. The apneustic area that is located in the lower pons will do the opposite to the pneumotaxic area. It transmits stimulatory signals to the inspiratory area that activate it and prolong inhalation. So whenever the apneustic area is active, the inspiratory area is active, so breathing will take long time or will be longer, so we have larger tidal volume. More air will be inhaled into the lungs. The result is a long, deep inhalation and a large tidal volume. When the pneumotaxic area is active, it overrides signals from the apneustic area. So if the pneumotaxic and apneustic areas are both active at the same time, the pneumotaxic area will override or will be the predominant area that controls the respiratory center or the inspiratory area. Now, what are the factors that regulate the respiratory center? The three different areas that are located in the medulla oblongata and the pons. The basic rhythm that is set and coordinated by the dorsal inspiratory area can be modified in response to inputs from different regions, like from brain regions, receptors in the peripheral nervous system, as well as other factors. So signals from the cerebral cortex, we can voluntarily alter respiration. For example, we can stop breathing for a short time, but this is only temporarily because the increases in carbon dioxide and hydrogen will eventually stimulate breathing. So we cannot stop our breathing for a long time. Finally, carbon dioxide and hydrogen buildup will cause us involuntarily to breathe or start breathing again. The other input or the other factors that can influence the respiratory center are chemoreceptors. Chemoreceptors are specialized receptors that sense changes in the chemical environment of the body, and input from central and peripheral chemoreceptors modulate the respiratory center, and we'll discuss the chemoreceptors in more details in the coming slides. Another factor or another input to the respiratory center are the proprioceptors. Proprioceptors are mechanoreceptors present in joints and muscles. So as soon as we start exercising, the rate and depth of breathing will increase because impulses from the joint and muscle proprioceptors will stimulate the inspiratory area. The other factor, number 4, is the inflation reflex, Herring-Brewer reflex, the names of the two scientists that describe this reflex. It is a protective mechanism to prevent excessive inflation of the lungs. When the lungs are overinflated, stretch receptors in the lung parenchyma are activated, which in turn inhibit the inspiratory and apneustic area. This will inhibit inspiration and therefore starts exhalation, and this is a protective mechanism. It prevents the breakdown of the alveoli due to overstretching. Other factors like temperature, pain, irritation of the airways, etc. So how respiration is chemically controlled? The chemical control of respiration, the respiratory system functions to maintain proper levels of carbon dioxide and oxygen. Therefore, it is very responsive to changes in the levels of these two respiratory gases in the body fluids, and mainly we are talking about the blood. The nervous system normally through homeostatic mechanism, adjusts the rate of alveolar ventilation almost exactly to meet the demands of the body, so that the oxygen pressure or the oxygen partial pressure and the carbon dioxide partial pressure in the arterial blood are hardly altered or changed even during heavy exercise and most other types of respiratory stress. As I said in the previous slide, there are two types of chemoreceptors. Chemoreceptors that are located in the central nervous system, we call them central chemoreceptors, located bilaterally in the medulla oblongata. They are highly sensitive to changes in hydrogen concentration and partial pressure of carbon dioxide. They are highly sensitive to changes in hydrogen concentration or partial pressure of carbon dioxide, or both, in the cerebrospinal fluid, and they in turn excite other portions of the respiratory center, which is present very close in the medulla oblongata. So the central chemoreceptors sense hydrogen and carbon dioxide concentrations in the cerebrospinal fluid that bathes the medulla oblongata. The peripheral chemoreceptors are present peripherally outside the central nervous system, located in the aortic bodies, in the aortic arch, and the carotid bodies at the bifurcation of the common carotid artery that you studied in anatomy. They are sensitive to changes in oxygen, so changes in oxygen concentration very strongly affect the peripheral chemoreceptors, and to lesser extent the partial pressure of carbon dioxide and hydrogen concentration in the blood. So they sense the changes of oxygen, carbon dioxide, and hydrogen in the. So location of the central and peripheral chemoreceptors. As you see here in this figure, we can see the inspiratory area in the dorsal part of the medulla oblongata. In the ventral part, or the surface, anterior surface of the medulla oblongata, we can see that we have the chemoreceptors, the central chemoreceptors. The peripheral chemoreceptors are located in the aortic bodies, in the aortic arch, and the carotid bodies that are present at the bifurcation of the common carotid artery. So let's discuss how changes in carbon dioxide, changes in oxygen, and changes in hydrogen can affect respiration or ventilation. Let's start with carbon dioxide. So an increase, we know that the normal partial pressure of carbon dioxide in the arterial blood is 40 millimeter mercury. This is what we studied in the previous lectures. So if the arterial partial pressure of carbon dioxide exceeds the normal value of 40, it is higher than 40 millimeter mercury, first it will weakly stimulate the peripheral chemoreceptors. Peripheral chemoreceptors will send input to the medullary respiratory center, and this will increase ventilation. Increasing ventilation will wash out carbon dioxide from blood. This will decrease arterial carbon dioxide partial pressure back to normal, which is 40 millimeter mercury. And this action on the peripheral chemoreceptors is weak. But the main action of carbon dioxide is its action on the central chemoreceptors. Carbon dioxide can cross the blood-brain barrier. It will enter into the CSF, or the cerebrospinal fluid, and there it will increase the concentration of partial pressure of carbon dioxide in the extracellular fluid. This carbon dioxide will bind with water in the presence of carbonic anhydrase, the same enzyme that we studied in red blood cells that help in the formation of bicarbonate, and this will lead to the formation of carbonic acid. Carbonic acid will quickly dissociate into hydrogen and bicarbonate. So this will lead to an increase in the brain extracellular fluid hydrogen concentration, which strongly stimulates the central chemoreceptors. When central chemoreceptors are stimulated, they will send input to the medullary respiratory center. This will lead to an increase in ventilation, wash out of carbon dioxide from the blood in the lungs, and this will lead to decrease in the partial pressure of carbon dioxide in the arterial blood back to normal, which is 40 millimeter mercury. There is another thing, that if the arterial partial pressure of carbon dioxide exceeds 70 to 80 or reaches very high values in the CSF, this will directly depress the medullary respiratory center. Therefore, the homeostatic mechanism, mainly through the central chemoreceptor and peripheral chemoreceptors, will try to bring carbon dioxide partial pressure back to the normal value of 40 before its level becomes very high and directly depresses the medullary respiratory center and causes death. Once arterial carbon dioxide partial pressure goes back to normal of 40 millimeter mercury, the feedback loop, or this concentration or this drop, will relieve the stimulus of this homeostatic or negative feedback system. As you can see here, that the effect or the mechanism that controls ventilation via carbon dioxide is an example of negative feedback system. The stimulus is high carbon dioxide partial pressure in the arterial blood, and the response is a decrease in the partial pressure of carbon dioxide back to normal. So partial pressure of carbon dioxide is the most important regulator of minute-to-minute respiration. That's why I started with the control of ventilation by carbon dioxide level. So the most important regulator of ventilation or minute-to-minute respiration is the level of carbon dioxide. Please put a red line about this most important. Partial pressure again of carbon dioxide is the most important regulator of minute-to-minute respiration, because changes in ventilation has immense effects on partial pressure of carbon dioxide. A very small change in ventilation can greatly affect the partial pressure of carbon dioxide in the blood. Peripheral chemoreceptors, as I said in the previous slide, are weakly sensitive to the partial pressure or changes in the partial pressure of carbon dioxide. The central chemoreceptors monitor partial pressure of carbon dioxide indirectly. As I said, carbon dioxide will be converted to bicarbonate and hydrogen, and the hydrogen will act on the central chemoreceptor. This means they do not monitor partial pressure of carbon dioxide directly, but the hydrogen that is generated from the carbon dioxide is stimulating the central chemoreceptor. And how? Carbon dioxide diffuses into the cerebrospinal fluid, which is one of the extracellular fluids, and combines with water to form hydrogen and bicarbonate in the presence of the enzyme carbonic anhydrase, as shown in the following equation. Hydrogen, or the formed hydrogen from this equation, stimulates the respiratory center and therefore increases ventilation. The more the carbon dioxide, the more hydrogen is produced and the more the ventilation is stimulated. Conversely, a reduction in carbon dioxide will cause reduction in hydrogen and ventilation will be reduced. As I said, when partial pressure of carbon dioxide is more than 70 to 80 millimeter mercury, this will cause direct depression of the respiratory center in the medulla. So we should avoid carbon dioxide to reach this level as doctors when we are in hospital and we are having a patient with high carbon dioxide in his blood. Now, what is the role of arterial hydrogen on ventilation? We saw in the previous two slides that hydrogen that is formed from carbon dioxide acts on the central chemoreceptors. But what about if there are changes in hydrogen in the arterial blood? There is a change in the arterial blood pH. Because hydrogen is positively charged, it cannot cross the blood-brain barrier. So be careful here. So it has no effect on the central chemoreceptors because it cannot cross or penetrate the blood-brain barrier. So any change in the arterial hydrogen, for example, increase in the arterial hydrogen, which is acidosis, this will stimulate the peripheral chemoreceptors, that in turn will stimulate the medullary center. It will increase ventilation, and washout of carbon dioxide will lead to a reduction in the arterial partial pressure of carbon dioxide, and this will decrease the arterial hydrogen. And this will relieve the acidosis or will correct the condition. Again, it is another example of negative feedback system. Arterial hydrogen has no effect on central chemoreceptors because it doesn't cross blood-brain barrier. Arterial hydrogen from carbon dioxide or other sources, and we are talking about hydrogen in the blood or in the arteries, strongly stimulates peripheral chemoreceptors and accordingly increases ventilation. So changes in blood pH, this change adjusts ventilation by its action on the peripheral chemoreceptors, not via its action on the central chemoreceptors, because hydrogen in the blood cannot cross the blood-brain barrier. Now, what is the effect of changes in the arterial partial pressure of oxygen on ventilation? Reduced arterial partial pressure of oxygen. We know very well that the arterial partial pressure of oxygen is about 100 millimeter mercury, and we studied that in the lecture of exchange of gases. When the arterial partial pressure of oxygen decreases to values less than 60 millimeter mercury, this will have different effects. Number one, it will strongly activate or stimulate the peripheral chemoreceptors, and the stimulation of peripheral chemoreceptors will stimulate the medullary respiratory center. This will increase ventilation, so we have more oxygen coming to the alveoli, more oxygen will enter into the blood. This will increase arterial partial pressure of oxygen. And peripheral chemoreceptors are sensitive to partial pressure of oxygen below 60 millimeter mercury because above this level, the percent saturation of hemoglobin with oxygen is 90%. If you go back to the oxygen or hemoglobin oxygen dissociation curve, you will see that in the range between 60 and 100 millimeter mercury, the partial pressure of 60 to 100 millimeter mercury of oxygen, the oxygen saturation is between 90 and 100%. So if the saturation drops below 90, then the peripheral chemoreceptors become activated. As you can see here, if we draw a vertical line, we can see that the saturation of hemoglobin with oxygen is 90 at partial pressure of 60 millimeter mercury. When partial pressure of oxygen drops below 60, this will stimulate the peripheral chemoreceptors. Nerve impulses via the glossopharyngeal and the vagus nerves will send impulses to the respiratory center. This will increase the rate and depth of respiration. This reflex is called life-saver reflex because at this lower partial pressure of oxygen, the respiratory center itself is directly depressed. Without these peripheral chemoreceptors, respiration would be depressed. So if the life-saver reflex doesn't work, this partial pressure of oxygen that comes to levels lower than 60 can directly depress the central chemoreceptors and the medullary respiratory center, and this can be life-threatening and can lead to death. Here we can see a summary of the influence of different chemical factors on respiration, and we are talking about carbon dioxide, oxygen, and hydrogen, both in the CSF and the arterial blood. So an increase in the partial pressure of carbon dioxide will weakly stimulate the peripheral chemoreceptors, but it will strongly stimulate the central chemoreceptors to stimulate ventilation. As I said, levels higher than 70 to 80 millimeter mercury can directly depress the respiratory area. An increase in the hydrogen in the CSF or the extracellular fluid of the brain has no effect on the peripheral chemoreceptors because this hydrogen is present in the CSF and cannot cross the blood-brain barrier to the blood. This high hydrogen in the CSF will strongly stimulate the central chemoreceptors, and this in turn will stimulate ventilation. An increase in arterial hydrogen, like in cases of acidosis, will stimulate the peripheral chemoreceptors but has no effect on the central chemoreceptors because, as I said, hydrogen cannot cross the blood-brain barrier. Low oxygen will strongly stimulate the peripheral chemoreceptors but only when its partial pressure drops below 60 millimeter mercury. And if it is not corrected quickly, it will directly depress the respiratory or the medullary respiratory area through its direct effect or direct inhibitory effect on the central chemoreceptors and also the medullary respiratory area. Here we can see the negative feedback system or the reflex loop or the feedback loop that controls the respiration in response to arterial carbon dioxide or changes in the arterial pH or oxygen. So the stimulus is an increase in the arterial partial pressure of carbon dioxide or decrease in the pH or oxygen in the blood. What are the receptors? The central chemoreceptors in the medulla, and these will mediate the effect of carbon dioxide indirectly via the generation of hydrogen. Also, these changes will affect or stimulate the peripheral chemoreceptors present in the aortic body and the carotid body. So nerve impulses will be sent to the control center, which is the inspiratory area in the medulla oblongata, which in turn will send action potentials to the effectors. These are the muscles of inhalation and exhalation that will contract more forcefully and more frequently. And this is called hyperventilation. The response is opposite to the stimulus, so there is a decrease in the arterial partial pressure of carbon dioxide, an increase in the pH, and an increase in the partial pressure of oxygen. This will return the homeostasis or return to homeostasis when response brings arterial blood, carbon dioxide, pH, and oxygen back to normal. Now let's talk about other factors. We cover the chemical factors, and these are the two respiratory gases, oxygen and carbon dioxide, and hydrogen that is closely related to the concentration of carbon dioxide. The other factors, as I mentioned in one of the previous slides, higher brain centers, they can modify breathing, either voluntarily or involuntarily. So I can stop breathing voluntarily or involuntarily, for example, when I speak, during singing, or emotional display and sleep. There is another system called the limbic system, which is also part of the brain, that is responsible for emotions. So emotional anxiety stimulates the limbic system, which in turn will stimulate the respiratory area. Pain can also affect breathing. A sudden severe pain will cause brief apnea. So it will stop breathing for a very brief period. Prolonged somatic pain will increase the respiratory rate. On the other hand, visceral pain in the internal organs, like in the abdomen or in the chest, will decrease the respiratory rate because ventilation will cause pain when we move the affected viscera by the inhalation and exhalation. So we try to reduce that pain by decreasing the respiratory rate. Temperature also can affect breathing or respiration. An increase in body temperature as when it occurs in fever, will increase the metabolic rate and increase the respiratory rate. On the other hand, low body temperature or hypothermia will decrease the metabolic rate because it will reduce the rate of metabolic reactions or enzymatic reactions, and this will lead to decreased respiratory rate. And this is logic because when we have low metabolic rate, we have low need for oxygen, and we don't produce that high amount of carbon dioxide to be removed from the tissues. Another factor that can affect breathing is irritation of the airways. Physical or chemical irritation of the pharynx or larynx brings about an immediate cessation of breathing followed by coughing or sneezing. Blood pressure was also found to affect breathing. A sudden increase in blood pressure can cause a decrease in the respiratory rate. Okay, let's go to some important definitions that you will encounter in the phase 2, especially in the tutorials, where you will get different clinical scenarios. So one term is hypoxia. Hypoxia means insufficient oxygen at the cellular level. So the amount of oxygen at the cell level is low, and this is called hypoxia. Hypercapnia is increased carbon dioxide in the arterial blood. Hypocapnia, which is the opposite, it is a decrease in carbon dioxide level in the arterial blood. Hyperventilation means increased pulmonary ventilation in excess of metabolic requirement. Dyspnea, it is difficult or labored breathing, and this is a very commonly used term to describe the symptoms or respiratory symptoms like in asthma, for example. Apnea, it is transient cessation of breathing. Respiratory arrest is the permanent cessation of respiration that happens during death. Cyanosis, the last term, is blue coloration of the skin resulting from insufficient oxygenated blood in the arteries. Please don't confuse yourself between hypoxia and cyanosis. Hypoxia, as I said, is low oxygen at the cell level. Cyanosis is blue discoloration of the skin and also mucous membranes that results from insufficient oxygenated blood in the arteries, which means, or usually it appears, when the amount of the oxyhemoglobin is equal or greater than 5 grams per deciliter. We study in the blood that the amount of hemoglobin, or the normal hemoglobin, varies between male and female, and it is usually above 12, and it can reach up to 18 in male grams per deciliter. But if 5 grams of that hemoglobin is deoxyhemoglobin that carries no oxygen, then we can see this blue coloration of the skin and the mucous membranes. Let's discuss the types of hypoxia. Hypoxia is a deficiency of oxygen at the tissue level, at the cellular level. Based on the cause of hypoxia, we can classify it into 4 types. Hypoxic hypoxia, it is caused by low partial pressure of oxygen in the arterial blood as a result of high altitude airway obstruction or pulmonary edema or fluid in the lungs. So the amount of oxygen in the blood is low, and this is called hypoxic hypoxia. So the blood already arrives to the tissue with low oxygen. The other type is anemic hypoxia. So the ventilation is fine in the lungs, the exchange is fine, but the transport of oxygen is low. Why? It is due to anemia, decreased amount of functioning hemoglobin in the blood. This will lead to reduced oxygen transport to the tissue cells, and causes include anemia, hemorrhage, etc. This is called anemic hypoxia. So the problem, we have less amount of hemoglobin carrying oxygen due to either anemia or loss of blood cells in hemorrhage or blood loss. Another type of hypoxia is ischemic hypoxia. In ischemia means low blood flow to tissue. So in ischemic hypoxia, blood flow to the tissue is reduced. So when there is low blood flow, it means less oxygen delivery to the tissue. And this happens when there is narrowing of the artery that supplies that tissue, either by atherosclerosis or by any compression from outside, any external compression on that artery. Finally, histotoxic hypoxia. The blood that is delivered to the oxygen contains enough amount of oxygen. There is enough amount of hemoglobin to transport oxygen to the tissue. The flow to the tissue is perfect, so the oxygen that is delivered to the tissue is not reduced, but the problem is that the tissue is not able to use that oxygen, that normal amount of oxygen that is delivered to the tissue. Blood delivers adequate oxygen to tissue, but the tissue or the tissues are unable to use it properly because of the action of some toxic substances. And the famous example is cyanide, because it blocks the enzyme cytochrome oxidase required for the use of oxygen during the process of ATP synthesis. So in histotoxic hypoxia, there is no hypoxia or hypoxic hypoxia, so the oxygen delivered is fine, amount of hemoglobin is fine in the blood, so oxygen transported well, the tissue perfusion is fine, there is no ischemia, but the problem is that the tissue cannot extract oxygen from the blood because of some toxins like cyanide. With this slide, we finish today's lecture. I hope you got the concepts. And if you have any questions, please don't hesitate to contact me by email. Thank you very much. And this is the last lecture of the respiratory system. Thank you again and wish you a nice day.